important process on the planet. If this didn't happen, without photosynthesis, not only is there no us, there is the diversity of life on the planet changes drastically, almost inconceivably. Inconceivable how it would change. You go to anaerobic conditions predominantly, and the diversity and the complexity of life on the planet would change. So it's not a hyperbole, it's not exaggeration to say this is the most biologically important process on the planet, both from the capture of energy. This is where energy that moves through all ecosystems begins, where the capture of it begins, and where the capture of carbon, for the most part, begins. The carbon that is in everything in this room that's carbonaceous, including us. It starts here with the capture, well, not in this particular diagram, but it starts with broadly photosynthesis and carbon CO2 fixation. So you can't overestimate how important this is. So it's certainly, you want to understand life on the planet, even at the most rudimentary level, it's important to spend time on it. So we left off somewhere around here. What we call the Z scheme, because it's kind of a Z on the side in terms of electrons moving around. But really, this is the photon capture, the energy capture processes. So we call two photosystems that work together. A couple things I want to reiterate. It's driven by light energy. That should be obvious, but it's important enough that it's worth repeating. And the transfer of that light energy, the capture of it, is by the pigment molecules, the carotenoids and the chlorophylls in the antenna complex of green tissue. That energy is transferred by resonance energy transfer to a specialized chlorophyll in the reaction center. So these guys aren't moving electrons around, these pigment molecules, here in the antenna complex. They're just channeling their energy from one to another through energy resonance transfer down to the specialized chlorophyll that's going to do the chemistry. It gets excited as it receives the energy, so the P680, as it receives energy from the various pigment molecules, it gets excited, and that electron that becomes excited from receiving that energy is donated into a short electron transport chain, right? So instead of just coming back, falling back to its ground state and releasing that energy as light or heat, some of which can happen, but this is where the chemistry begins, right? But because it's donated an electron into an short electron transport chain, it has to get electrons back. Otherwise, it's done. It has to keep the process going, it has to get electrons back. That's what water's for. So remember, all these pathways and processes in metabolism, I try not to get bogged down in the names of the electron carriers, the plastoquinones and plastocyanins and such. But you're asking the question, what's it for? What goes in, what comes out? How is it regulated? And where does it take place? This takes place in the chloroplast and the thylakoid membrane. That's why you want all that large amounts of membrane surface area. So we talked about that on Wednesday. So on the thylakoid membranes, in green tissue, in chloroplast, what's it for? Well, that's obvious. You know this. You've known this since you're a little kid. It's to capture light energy, and then we'll move to a slightly bigger picture, and store some of that as potential energy in the form of chemical bonds, first creating electrochemical gradients, just like in the mitochondria. So as we go through this, draw parallels and so compare and contrast, if you will, the mitochondrial activity and chloroplast activity. There's lots of similarities and there's some differences. One of the differences is in the what goes in, what comes out. So water goes in here, which is the opposite of mitochondria. Water comes out of that mitochondrial activity because oxygen is the terminal electron acceptor in the electron transport chain in mitochondria. Here, so you're reducing water. Here you're oxidizing water. So loss of electrons, oxidation. Water is losing electrons. That's why plants want water in this process. Water does lots of other jobs, obviously, in plants and all cells. Solutes, participates in reactions. But here it's donating electrons. It's getting oxidized. Those electrons return electrons to the P680 that's donating an electron into the chain, so you can keep it going. You can keep it going as long as you keep putting energy into it in the form of light. Okay, so that electron now goes from one carrier to the next. coming back down in energy level. Some of that energy, as we'll see in the next slide, can be used to do work, move a proton against electrochemical gradient. But it's donated to photosystem 1, to specialized chlorophyll in photosystem 1. Why? Well, photosystem 1 needs an electron because it's been excited in the same kind of process. It's been excited. So what isn't shown here, you should annotate in your notes or something, is the little lightning bolt. So light, they're showing energy transfer, resonance energy transfer here. But of course, these got excited by light. And it's the same process. The antenna complex, the light-absorbing pigments have been excited by light. They've transferred that energy around in the complex, in the antenna complex, ultimately to a specialized chlorophyll, a little bit different than the one in photosystem 2. Same process. When it gets excited by that energy resonance transfer, that is a consequence of light excitation by light absorption, it donates an electron into a short electron series of carriers. So it's donated an electron. So it has to get one back to keep the process going. Where is it going to get that electron? It's not getting them from the pigments. These are not donating electrons. They're just transferring energy from one pigment to another through that resonance energy transfer. So it gets the electron back from photosystem 2, from the pool in the electron transport chain. It gets that electron back so you can keep going. But it has to have light. That's how these got excited in the first place. So you have to be putting energy into it because you're moving an electron to a higher energy state. Right? It's not explicitly shown in this diagram, but hopefully this is a higher position than these because it's at a higher energy level. And then you're going down in energy level, and then you're going back up. But if you're going to go back up in energy, you have to put energy into it. That's the light coming into photosystem 1. Okay, so just one aside. And this is roughly where I left off on last week. But I wanted to revisit all that. It was Wednesday. The oxidation of water takes place at photosystem 2. That's where it gets electrons. Oxidation of water. That's where it gets electrons. Those protons are useful, and we're going to see that in a second, for establishing that potential energy gradient, right? So electrochemical gradient. No small task to split water. You can have water sitting in a beaker, and you're not going to get a whole lot of oxygen evolution of that water unless you put a lot of energy into it to hydrolyze. It's a pretty stable molecule. But this manganese cluster at the reaction center is impressive. It's able to split water, and the protons are released. We'll see how we'll use them in a second. And the oxygen is essentially a byproduct. So we don't really talk about oxygen as a product of photosynthesis. From the plant's view, it's not really a product. It's a side, made some oxygen. Maybe the mitochondrion captures it. Maybe it just dissipates. It's important to us, important to the rest of the planet. But from the plant's point of view, that oxygen production is not really an end goal here. It's establishing the electrochemical gradient, ATP synthesis, NADPH production. So this is why plants, one of the reasons they need manganese, just like we need manganese, for certain enzymes. Right? So we looked at that as cofactors, or metals as cofactors a few lectures ago. So now you have this parallel. You've already seen some differences with the mitochondria. You see some structural similarities in lots of membrane, inner membrane. So for the ETC on mitochondria, for thylakoid disc light capturing in chloroplast, you see some differences, oxidation versus reduction of water. It's reduction of oxygen to give you water in mitochondria. It's the oxidation of water to get electrons. Mitochondria, the electrons start in glucose, or whatever, it could be fats, but let's talk about glucose. The electrons start there, they end up on oxygen to give you water. Here, the electrons start on water, so you're essentially going the other way. The electrons start on water, and they end up, at least initially, temporarily, an NADP to give you NADPH. Well, how do they get there? So we just saw them going from photosystem 1 into a short electron transport chain. That will move electrons to, where's my cursor? There it is. So it's been energized. Here you see both the light exciting both photosystem 2 and 1. Electrons go into this short chain for the second round, if you will, of electron transport, and they end up at the NADPH oxidase, reductase. You're reducing NADP. So NADP, remember, is the low energy form of the coenzyme. NADPH is the high energy form. The point, back to the what goes in, what comes out, and what's it for, the point is to capture some of the light energy, some of the energy in light in high energy compounds, ATP, NADPH. How is that achieved? Well, one way it's achieved is by using light to excite electrons and then moving them through a series of carriers, ultimately to a coenzyme to give you NADPH. So you reduce it, it gains electrons, now it's at a high energy state. And to use some of that energy as those electrons move from one carrier to the next, ultimately to NADP, to use some of that energy to generate and maintain an electrochemical gradient, right? So this is the proton electrochemical gradient. As the electrons are moving down through these carriers, you can, just like in the mitochondrion, so here's a parallel, use some of that energy as you go from one carrier to the next, from higher energy to lower energy. Instead of just releasing that energy as heat or light, use it to do some work. Move protons against their electrochemical gradient into the thylakoid lumen. So the pH is going to drop. Positive charge is going to go up. That's a gradient. Gradient represents potential energy. It's like water behind the dam. And just like another parallel here, the mitochondrion, you can use that potential energy, convert it to kinetic energy, spinning the ATP synthase, because those protons, in quotes, want to go down their electrochemical gradient. So you give them a port to do that, but that port, that hole, is an ATP synthase, and as they move through there, spins that turbine, just like water coming through the turbine in the dam, kinetic energy, captures some of that kinetic energy back in the form of bonds, covalent bonds, to drive that endergonic reaction of ATP synthesis. So ATP and NADPH come out. We see how they come out. We know we're going to look at what they're for in a second. But there's the purpose of the light reaction, to capture some of the energy and light in these high energy molecules, NADPH and ATP. Yeah, question?
If the plants didn't have like a mitochondria, could they survive just with the energy they get out of photosynthesis?
It's a very good question. So the question being, can you have plants without mitochondria? Because maybe they don't need this second powerhouse. In general, no. Because the mitochondria serves other biosynthetic reactions as well that are important in plants, and in what we call dark respiration, so respiration at night, plants will be using their mitochondria for metabolic energy. But they're less reliant, for sure, they're less reliant on the mitochondria as the energy powerhouse. They're reliant on chloroplasts, right? Okay, so we've got where it happens, thylakoid membranes for the light reactions. We're going to look at the carbon fixation reactions in a second. What goes in, what comes out. How is it regulated? This is regulated predominantly at the level of light availability. But in addition to that, the cell will regulate the light reactions as it judges its energy state. So cells, broadly speaking, monitor their energy state, and they do that by looking at the ratio of high energy compounds to low energy compounds. We won't go into the mechanisms of it, but it's fairly well studied and established. So if a plant cell is saying, well, you know what, I need some ATP, but I don't need any more NADPH. I have enough of that high energy compound. It can adjust the activities, the relative activities of the two photosystems, and instead of sending the electron to NADP reductase, so into that part of the chain, photosystem 1, it can send the electron back into the sort of general, the pool, if you will, the chain in between the two photosystems. And this is called cyclic electron transfer. So instead of sending, when that specialized chlorophyll is excited, instead of sending that electron ultimately through a short chain to reduce NADP, it sends it back to the chain between the two photosystems and just cycles it. Obviously, you would have had to start with some electrons from photosystem 2, from water, but once it's in there, you can keep it cycling. And if you're keeping it cycling, you'll have a source of protons, There's always a source of protons in a cell. And you can maintain your electrochemical gradient, so you can keep ATP synthesis active. Right? So it's a balance. Cells always have to be looking at their metabolic activities and judging what do I need to speed up, slow down. And so we've looked at this in a number of contexts now. So here's just another one. So be comfortable comparing the oxidative phosphorylation, that's mitochondrial activity, phosphorylation of ADP. It's oxidative, it needs oxygen. And the photooxidative, it's light driving that oxidation ultimately of NADP. Okay, so you know what goes in, what goes out. What comes out? Water goes in as a source of electrons. It's not glucose, a source of electrons, unlike mitochondria. And we looked at the flexibility of mitochondria on Wednesday. You can send other carbon skeletons through those pathways. You got water as your source of electrons. What comes out? Well, oxygen is a byproduct, but small energy-rich molecules, ATP, NADPH, come out. Okay, what good are they? Well, you know they're high-energy compounds. They can drive endergonic reactions because their products are so much lower energy than themselves. ATP is a much higher energy level than its products, ADP and phosphate. And when you look at how much energy it takes to bring inorganic carbon into an organic form, that's what you need the ATP and NADPH for. So look at 6 carbon, so glucose is a 6 carbon molecule. If you want to make a sugar, that's how much energy you have to put into it. That's a huge amount of energy. But that should be no surprise, because you know if you go from glucose all the way down to CO2 and water, that's the bottom of the hill. Top of the hill to the bottom of the hill, basically. So if you're going the other way, you've got to put a lot of energy into it. So NADPH, ballparkish, each one of them is worth, in quotes, a couple of ATP. So it's a huge amount of energy. All right, so the point of the Calvin-Benson cycle, again, approach it the same thing. What's it for? What goes in, what comes out? How is it regulated? Where does it take place? Let's start with the simple kind of memorization part. It takes place in the stroma of chloroplast. So that's the liquid that surrounds the thylakoid membranes. It's kind of functionally analogous to the mitochondrial matrix. Right? So these are soluble enzymes. They're not embedded in membranes. Probably some are membrane associated. But in general, we're thinking of the liquid surrounding the thylakoid discs. That's where the ATP, if you go back a slide, that's where the ATP and the NADPH wind up. Perfect. That's where they're needed. But you got to bring inorganic carbon into the system. This is where on the planet you go from inorganic to organic carbon, and that organic carbon then spreads through all the trophic levels, all the ecosystems, and ends up everywhere you look at carbon that's in some organic form. But it has to come into the plant through these portals. So these holes, these are the stomatopores on leaf surface, and they open and close according to the plant's need for CO2 to come in and water vapor to leave. We don't look at so much of the role of water vapor coming out of here. It's important in vascular tissues and plants and the movement of materials. Don't really cover it in this course. Isn't time to. But a plant, this is a very important point, we'll come back to in a minute. A plant has to make a decision on when to have these open. See this in the left panel here, and closed. You see this in the middle panel. Because it's a balance. Let CO2 in, but water is going to escape. Parasites also, and pathogens also exploit this hole. Fungi, pathogenic bacteria. This is it's just a doorway inside the leaf. So it's controlled, and these are the guard cells that control that. So these two sort of together as a lip, shaped like a lip, and their opening and closing is a result of tonicity changes. Pull ions into them, water will follow, they'll swell up and open. Move ions, solutes, let's just say solutes out of them, the water will follow, they'll become flaccid and close. This is a huge decision for a plant. When to be open, and if the plant has to make the decision, oh, I need CO2 for photosynthesis, but if I open up, I'm going to lose a lot of water, it's dry and hot, and I don't have a lot of water in reserve, it will choose closing every time. There's always some storage energy reserve you can go to, but if you lose too much water, there's no going back often. Right? So even though it looks structurally simple and okay, this is a hole that CO2 comes in, water goes out, huge decisions from a plant's perspective. Right? This is where the carbon becomes organic on the planet. These notes are just for your reference. Let's look at what happens. Don't get bogged down in the names of the structures. Don't get bogged down in the names of the enzymes, except for the main one at the top there, rubisco. So we know where it happens. What goes in, what comes out? CO2 goes in. High energy small molecules, ATP, NADPH, go in. And it's regenerative. So it regenerates the carbon acceptor, the 5-carbon acceptor. That one you should know the name of, ribulose bisphosphate. So 5-carbon high energy receptor, it's dual phosphorylated. Makes it very reactive to add a CO2 to it. So organic carbon, inorganic carbon coming together to give you a 6-carbon compound, 5 from the ribulose, 1 from CO2. And that 6-carbon compound is very unstable. Those phosphate groups make it less stable, high energy. Splits into 2 3-carbon compounds. And once you've run this cycle a few times, you can start to siphon off some of that 3-carbon product. You'll use some of it to regenerate your 5-carbon molecule to keep the cycle going. So in that respect, it's similar to the Krebs cycle. But it's really only similar in that it's regenerative. The Krebs cycle releases CO2 as a byproduct. This is a reductive cycle. The Krebs cycle is oxidative. You're oxidizing carbon. You're taking electrons off of carbon in carbon-hydrogen bonds in the Krebs cycle. Here you're putting electrons onto, you're reducing these carbon skeletons. So they're opposite in that respect. So you can start to siphon off 3-carbon compound, very versatile building block, and kind of leads the way for everything else. Just let that sink in for a second. All the diversity, the thousands of different carbon-based molecules you might have in any given plant, and then the hundreds of thousands of different carbon-type molecules you may have on the rest of the planet, start here for the most part. Not quite that simple, but you could certainly make a case for it. Right? So it brings organic carbon into the biological world. Right? So that's what it's for. What goes in, what comes out, these high energy compounds. So a good 3-carbon building block. And it's regenerative. Where does it happen? In the stroma. How is it regulated? It's predominantly regulated to some level at substrate availability and energy availability. If the ATP, NADPH is low, you can't run it. If the CO2 there isn't enough CO2 substrate, you can't run it. But rubisco itself, the most abundant enzyme on the planet, and again, you could argue the most important enzyme on the planet, bringing organic carbon into the organic world, it is regulated at a few different levels. We won't go into the details of the regulation, but that's the key regulatory point, is the regulation of rubisco itself. And of course, because it's tied to energy metabolism through ATP and NADH, it's regulated by the energy status of the cell. All right. So we do talk sort of about different phases. Where's my carbon fixation phase, reduction phase, and regeneration phase? Right? You have to put some energy back, additional energy into it for that regeneration. So you need energy for the reduction, you need energy for the regeneration. It's an energy-greedy process. CO2 and water all the way back up to a complex carbon skeleton, or en route to complex carbon skeletons. A lot of work. Okay. The weird thing about rubisco, most abundant, most important enzyme on the planet, yet it's strangely inefficient, it seems. It's a conundrum that scientists have it's kind of baffled scientists for a very long time. And there's still an active debate about it. Because it's rubisco. Ribulose bisphosphate carboxylase oxygenase. It's natural, if you will, substrate is CO2. But oxygen is an alternative substrate. If oxygen gets into the active site, the enzyme can catalyze the reaction. And you get one useful product and one product that is of questionable use. In fact, so that's called photorespiration, because it's respiration in the sense it's using oxygen. And in order to get that product, when oxygen is the substrate, in order to get the product, the 3-carbon product, back into primary metabolism, really, it takes about 4 different compartments in the cell and something in the order of 12-ish reactions, and some energy, and some carbon is lost. It's a really complex process, photorespiration, and it seems wasteful on the face of it. You do the calculations, and you can grow plants under oxygen-poor conditions versus CO2-rich conditions, and they do, no surprise, way better, most plants, C3 plants, with high CO2, low oxygen. You don't need the mitochondria that much. So if this is wasteful, scientists have estimated that it's roughly, you can lose 30% productivity. Imagine your crops, 30% more productivity. That's mind-blowing amount. That's massive. Most farmers are happy a few percent. You promise a canola farmer 2% more oil, that's huge money. 30% productivity, that's hard to wrap your head around that much. So why is it there? Why is photorespiration there? And so this is the conundrum. It seems wasteful, and people have tried to make biotechnically better Rubiscos. If you could engineer in the lab a better Rubisco that didn't bind oxygen, but had a great preference for CO2, you would probably have not just $1 billion product on your hand, but a great way to increase the productivity of food for the planet. So far, there's been little to no success in that front. So some scientists argue it's actually important. It evolved, it's not a hangover. So some scientists argue it's a hangover because Rubisco evolved in an oxygen-poor environment in the very early days of the planetary, geologic evolution, if you will. So there was not much oxygen around. It wasn't a problem. And it's somehow constrained that you can't make it better. If you make it better at choosing CO2, it's less efficient or some other cost to pay. Others say, no, it's important because it's an overflow energy valve. If the cell's got too much energy, this is one way to deal with it. Waste some energy with photorespiration. And still there's a third argument that says, no, it's important for nitrogen metabolism. Because if you mess around with photorespiration, there's a price to pay with nitrogen metabolism. There's a balance between carbon and nitrogen metabolism. It's still an unresolved argument. And yet, so that long segue, but hopefully you found it somewhat interesting. And yet, how important can it be if you have plants on the planet that have evolved fairly recently and independently, different families of plants, ways to get around it with C4 metabolism. So we call what we just looked at, the Calvin-Benson cycle, C3 metabolism, because the first product of that carbon fixation, first real stable product, is a 3-carbon compound. C4 metabolism, the first stable product, is a 4-carbon compound. And C4 plants use a different enzyme, a better enzyme, PEP carboxylase. It's not found in animals. We don't have it. It's great at grabbing CO2. It has no problem with oxygen. It doesn't get confused. Is this my substrate? Is this my substrate? It has no oxygenase activity. So when it grabs the CO2, it gives you a 4-carbon compound. It takes a 3-carbon compound, high-energy compound, PEP, phosphoenolpyruvate, puts a CO2 on it, gives you a 4-carbon organic acid. That all takes place in one type of cell, mesophyll cells in the leaf. Then that 4-carbon organic acid is shipped to a different cell. So this is a spatial segregation. That's a phrase you should know. Spatial segregation, so different cell where Rubisco is, where the Calvin cycle is. It sends that organic acid to that other cell. Now it decarboxylates it, chops that CO2 off it, and saturates the environment around Rubisco with CO2. So now Rubisco doesn't have to worry about oxygen. You have a very efficient use of energy, basically a much more efficient carbon capture. So these are more productive. Less carbon is wasted, and in addition, they're better at conserving water because they don't have to have their stomatopores open as often because they're more efficient with CO2. They can have them closed more frequently. You close your stomata more frequently, you don't lose as much water. You don't lose as much water, you're better at utilizing water. You have better water use efficiency. And obviously, agronomically, that's tremendously important. But even just ecologically, period. If you're a plant, water resource management is tremendously important. So that's called C4 metabolism. So they've solved the problem of loss of carbon, of inefficiency. They still use Rubisco, but the first carbon capture step is by PEP carboxylase. So that's definitely an enzyme you need to know the name of and its role. Okay, so spatial segregation. But you can also do a temporal segregation. Independently, ways to avoid photorespiration have evolved in in other families of plants. It was first found in the Crassulaceae family. So these are mostly succulents and xerophytic plants, meaning they grow in dry, arid, hot environments. And what do you expect if you're in that environment? You need high water efficiency. So these are the most water efficient plants. So the CAM plants are more efficient than C4, that are more efficient than C3. Why are they so water efficient? In one cell, I'm not crazy about this diagram. This is one cell. They've solved the oxygenase problem temporally. C4 plants solved it spatially, they solve it temporally, so time. So at night, you're living in the desert. In the day, the sun is blasting on you. There's very little water around. You do not want your stomatopores open. You're dead. They close those stomatopores in the day, and then so they're not losing water. And at night, they open up the stomatopores when it's cool, and they're not getting blasted with radiation. Let CO2 in. They use PEP carboxylase at night to fix that carbon into an organic acid, a 4-carbon organic acid. So it's the same reaction, PEP carboxylase. And then when the night ends, they close their stomatopores again. They've stored that organic acid all night, and in the day they liberate it from the vacuole and decarboxylate it in front of Rubisco in the same cell. So they solved that problem. So now there's very little oxygen around relative to CO2. You're constantly decarboxylating organic acids in front of Rubisco. It's going to have no problem. There's going to be little to no competition for the oxygen, or for the active site between the oxygen and CO2. And you've also solved the water use problem. Yep?
Since CAM and C4 both use PEP carboxylase, does that mean that there's like an evolutionary connection between those two groups?
So the question being, is there an evolutionary connection between them both using PEP carboxylase? Plants have PEP carboxylase. Microbes have PEP carboxylase. We don't. So historically, in the bacterial PEP carboxylase has some evolutionary relationship to plant PEP carboxylases. So it's in the ancient lineage before plants evolved. I'm not sure if that answers your question. But they use PEP carboxylase plants in two different contexts. In the photosynthesis context, in C4 and C3 plants, in C4 and CAM plants. But they use PEP carboxylase elsewhere. Even C3 plants use it, but not in photosynthesis. Not sure if that's over answering your question. Okay, so you've solved two problems. Waste of carbon, because of the oxygenation, and water use, which is obviously, if you're in an arid environment, extremely important. But one thing I forgot to point out, in both C4 and in CAM, is that there's an extra energy cost to this. Because you have to regenerate that high energy carbon receiver. So there's all the energy cost of Calvin cycle, of course. You can't get around that. You're turning CO2, oxidized carbon, into a reduced form of carbon. But you also have to regenerate that phosphoenolpyruvate. That's a high energy molecule. You have to put ATP into it. So these plants tend to grow where there's higher light energy environments, so typically more tropical environments. A lot of them are really fast-growing grasses, tropical grasses like corn and sugarcane. These are, like I said, mostly plants in dry, hot environments. Not exclusively, certainly. There's CAM plants around here. But they've solved the same problem in two different ways, spatial versus temporal. But wait. Okay, so if you're decarboxylating You're releasing CO2 in front of Rubisco. This is all in one cell. PEP carboxylase is in that cell. PEP carboxylase is way better at grabbing CO2 than Rubisco is. So why doesn't it just grab it again? And you go through this futile cycle. PEP carboxylase grabs it, then there's a decarboxylase that chops it off, PEP carboxylase grabs it again, and so on. And you would have that futile cycle because it's all in the same cell. You don't have that concern in C4 because you've got them in two different compartments, well, two different cells. You would have that problem here, potentially at least, but what happens is the cell diurnally regulates, so with a circadian rhythm, regulates the PEP carboxylase activity, shuts it off in the day, turns it on at night. And that avoids that futile cycle, both through course regulation of synthesis degradation and also through post-translational modifications. So that's an important aspect. Okay, so this is a slide just kind of comparing these strategies. Independently evolved to solve the same problem. But there's still lots of biotech interest in trying to figure out, can we make a better Rubisco? Can we turn soybean, a C3 crop, into a C4 crop? That's tricky because you got the anatomy difference there, two different specialized cell types. Can we turn soybean, a C3 crop, into a CAM plant? Can we put PEP carboxylase in there and regulate it diurnally? And there's lots of interest in achieving these kind of biotechnical goals. So again, back to this diagram. This is a great diagram to look at, not to redraw, but to look at and say, do I understand? Where the processes take place? What goes in, what comes out? How are they regulated? Right? So those are the main things. What are they for? What are they for? What's the purpose of the carbon carbon fixation reactions? That one's obvious. Bringing inorganic carbon into the organic world, starting off and generating a small carbon skeleton that's a very good building block. Oxygen, just a byproduct. Electrons start in water, quite different than where the electrons start in the primary pathways we looked at from glycolysis, etc. So parallels and differences. So here's some of the these take-home messages. You should be able to look at these and expound on them, intelligently. I was going to roll into chapter 11, but I'm going to stop there. I'm going to post chapter 11 as an independent, just as a video you can go and watch. It's fairly short. It's about 38 minutes long. This is the last lecture that I actually deliver. And the last time you guys will see me in this course, no, except for Wednesday, we have the in-group quiz. Sorry, in-class group quiz. I'll take, because I have some time, I'll talk about that here. But I'm not delivering a lecture, so this is it. But Wednesday, you come in, we'll have 2 questions. They're challenging questions. You can use your notes, you can use the web, you can do anything short of sending that question to someone else. These aren't Googleable questions. You can do it by yourself if you want. You can join a group, discuss it with them, submit questions. Everyone submits their answers independently, but you can make your decisions based on the group discussion, or you could join a group, disagree with them, and say, well, okay, you guys think it's C, but I'm pretty sure it's B. I'm going to go with B. So this is an opportunity to solve problems together, to consult, which is more realistic in the world. You're rarely having to solve problems without any access to any kind of resource. So that's what makes most exams kind of a false situation. But there are questions along the lines of, I'll give you a scenario, describe with some description, maybe some data, and it's a problem to solve. Describe why these 3 mutants look like this, why this product is built up and this one is low levels of it, and so on. And I'll give you 5 hypotheses to choose from, 4 or 5, which ones make the most sense. And they're not easy. They'll take you the full 20 minutes for each question. But we've gotten good feedback from it. It's a real good test of understanding the material and an opportunity to problem solve as a group. All right, so that said, we're done now. Have a good weekend. Have a safe holiday weekend, and I'll see you guys next Wednesday.